Greetings. Welcome to the second lecture in Sealaska Heritage Institute's lecture series entitled, We Have Lived in Southeast Alaska Since Time Immemorial. I am Chuck Smythe, Director of the Culture and History Department. SHI has started construction of its arts campus in downtown Juneau, which will expand opportunities for education and art. If you are interested in making a donation to the arts campus, please visit sealaskaheritage.org/campus. The title of today's lecture is Paleogenomics, Community Engagement, and Evolutionary Histories of Indigenous Peoples of Southeast Alaska and the North Coast of British Columbia. As you watch the lecture, we invite you to submit questions in the chat box in YouTube. Professor Malhi has graciously agreed to answer questions at the end of his presentation. Professor Ripan Malhi is Romano Professor in the Department of Anthropology, School of Integrative Biology, American Indian Studies Program, and the Carl R. Wuss Institute of Genomic Biology at the University of Illinois, Urbana-Champaign. He earned his PhD from the Department of Anthropology at UC Davis and then completed a postdoc in the Department of Human Genetics, University of Michigan Medical School. Prior to starting at the University of Illinois, Professor Malhi co-founded a biotechnology company called Trace Genetics in the San Francisco Bay Area. He is a molecular anthropologist who collaborates with indigenous communities to study the evolutionary histories of indigenous peoples of North America. Professor Malhi co-directs the summer internship for indigenous peoples in genomics, an NIH-funded program for indigenous community members to learn innovative concepts and methods at the cross-section of indigeneity and genomics. He is also the co-director of the IDEAS program called Increasing Diversity in Evolutionary Anthropological Sciences, and executive director of the journal Human Biology. Professor Malhi.
Great, thank you for that nice introduction, Chuck. I'd also like to thank you, Dr. Zito Worrell, and the committee for putting together this lecture series. I was able to sign in and watch Dr. Dixon's lecture on Tuesday, and I really enjoyed it. So I also want to acknowledge and thank the communities associated with Sealaska Heritage Institute, so the Tlingit, Haida, and Tsimshian communities. I'm not in Juneau. I am actually zooming in from central Illinois at the University of Illinois in Urbana-Champaign. You can see my background here is a photo of the quad and some of the buildings on campus during the summertime. The University of Illinois was founded in 1867. And 150 years later, now the university is starting to reach out to communities that used to be on these lands. And so I would like to acknowledge these communities as well. The Peoria, Kaskaskia, Piankashaw, Wea, Miami, Muscooten, Odawa, Sauk, Meskwaki, Kickapoo, Potawatomi, Ojibwe, and Chickasaw nations. These are their traditional territories before they were forcefully removed before the founding of the University of Illinois. Okay. So let's start the presentation. So today I'm going to be talking about some of our work on paleogenomics and how we use community-based methods with indigenous communities in these regions and partner with them. Just to give an outline of the presentation. So the first half is mostly going to be on how our efforts to help indigenize science through the SING program and also through community-based methods. And then it's the second half where I'm going to be talking more about the paleogenomic projects and partnerships with Indigenous nations. So there's a long history of disrespectful and harmful actions of scientists, by scientists with Indigenous communities, from when Europeans first came to these lands up until today. So I wanted to start by just talking about some of these case studies that focus on genetics. So the first case is with the Havasupai tribe, and this was in the 1990s. So the Havasupai members have a higher than normal or just a high incidence of type 2 diabetes. And so there were researchers at universities in Arizona that were studying the blood samples from Havasupai community members in order to identify the genetic underpinnings of type 2 diabetes. So the researchers were unfortunately misused the samples by using the samples for other studies of disease, as well as other studies not relating to health at all, on origins of Indigenous peoples in the Americas, without the knowledge of the Havasupai. When the Havasupai found out that the samples were being used for other studies, they filed a lawsuit with the Arizona Board of Regents. There's another case that happened in the 1980s, about a decade earlier, but instead of in the Southwest, this was in Canada on Vancouver Island, where again, a researcher, genetic researcher, was studying blood samples from Nuchalnoth community, because the Nuchalnoth had a high rate of rheumatoid arthritis. And so this researcher was trying to identify genetic underpinnings of rheumatoid arthritis. So after the researcher went and collected the blood samples and left, the researcher did not return to the community to report end results. And the researcher, after finding negative results, ended up using these samples again for other purposes without the knowledge or consent of the community. And so that was another example of misuse. And then the last example that I want to discuss is more with paleogenomics. On the banks of the Columbia River in 1996, an ancestor was eroding out of the banks, and the local Indigenous communities wanted to repatriate and rebury this ancestor right away. Unfortunately, they were not able to, because scientists filed a lawsuit to block the repatriation, because this ancestor was nearly or over 8,000 years, believed to be over 8,000 years and when it lived, and had a near complete skeleton. And so the scientists thought that this was a national treasure, is what they claimed, and wanted to study this ancestor. And so it wasn't until about 20 years later that the Indigenous community members were able to repatriate and rebury this ancestor. And this 20-year period was very distressful for the local Indigenous communities. And so these case studies are some of the examples of why science and the history of science provide an unwelcome environment for Indigenous, might provide an unwelcome environment for Indigenous community members. And so there is a very small number of Indigenous scientists in general, and to point to a specific example, here is some data from the American Society of Human Genetics membership in 2017. And this is typical of most scientific memberships or institutions, where you can see that a majority of the scientists are white, and then there is a small percentage of minority scientists. And here you can see that Indigenous peoples show a very small percentage of 0.15% under the American Indian, Eskimo, and Aleut category right here. So there's a need for more Indigenous scientists. And in general, there's a need for more inclusive science. And so we have efforts to make science more inclusive through the SING program, and then also give Indigenous peoples more of a say in the research process by using community-based research methods. And so first I wanted to talk about the SING program in general before going into our community-based methods. So the SING program is a program that features a 1-week workshop that is usually held over the summer, as suggested by the name. And it's a program where we bring in 15 to 20 Indigenous scientists or students or Indigenous community members, where the scientists or students are early career. And we discuss the crossroads between indigeneity and genomics research, and discuss how we can use genomics as a tool to help Indigenous communities in things that they need. And so it's a workshop that is unique and that it is mostly held by faculty who are either Indigenous scientists and scholars or allies like myself who work closely with Indigenous communities. And so here is a photo of the faculty of the 2019 SING workshop that took place at the Institute, Carl R. Woese Institute for Genomic Biology at the University of Illinois. And so the way the workshop is run for the week is it has different modules. And so one of the modules that we have is a molecular biology module where participants can extract their own DNA from a saliva sample if they wish to, or they can observe someone else extracting the DNA so they can see the process. And then once they extract the DNA, they sequence a portion of the mitochondrial genome and do an ancestry analysis. And so after sequencing a portion of the DNA and doing the analysis, we have a session on reductionism and how identity is not only a result of biology, but also just one component of multiple components of a person's identity. And so we talk about biological reductionism a bit. We have another module that is on bioinformatics. And for this computational genomics and bioinformatics module, participants are able to analyze hundreds of thousands of DNA markers to do analyses on human genetic variation. We have lectures and discussions on how genomics can be used as a tool within an Indigenous framework by leading scholars. But a majority of the time is spent in large group or small group discussions. And CELSI stands for Cultural, Ethical, Legal, Social Implications of Genomic Research with Indigenous Communities. And so we have discussions about how genomics research can impact communities, in this case, Indigenous communities. And it ends up being kind of a brain trust where we review past ideas and build on them. And so some of the ideas that we discuss and build on are concepts like the DNA on loan idea that was originally introduced by Arbor and Cook in 2006. And this idea is that a sample given by a participant from an Indigenous community is not owned by a university or the researcher that takes the sample, but the researcher is a steward of the sample to care for it. But the DNA sample and the biological sample still belong to the participant and the community that they came from. We also talk about how once genetic data is generated for a community, it is the researcher's responsibility to make sure that that data is not misused in any way. And so I like this quote by Dr. Kim Tallbear dealing with this idea that if you're going to work with Indigenous communities on genetics, you will have to be willing to make lifelong relations. We have other exercises in the workshop. Here is a photo of a fun exercise that we have, kind of showing how you can indigenize the lab. So we have taken white lab coats, and we are de-whiting these lab coats by having participants bring in their culture and their experiences and decorate the lab coats with those experiences and culture. So the SING workshop in the USA has been happening since 2011. And we've had 7 workshops so far. And so far, overall, we've had over 120 participants from all across North America. A number of the workshops have taken place at the University of Illinois, but then there are other workshops that have taken place at other places, such as in 2014, the University of Texas at Austin. In 2017, we had the workshop at University of Arizona in Tucson, and in 2018, it was at the University of Washington in Seattle. So since we started the SING workshop in the United States, the workshop has been organized and has begun in other places around the world. For example, in 2016, Maui Hudson and Phil Wilcox and others have started the Sing Aotearoa workshop in New Zealand. In 2018, Dr. Kim Tallbear moved to University of Alberta and started the Sing Canada workshop with Jessica Kolopenik and others. And in just last year, or in 2019, Sing Australia was started. There was plans for a Sing Mexico in 2021, but because of the pandemic, that looks like it's being delayed till 2022. And there's interest from other local communities in starting up a workshop like this. So in 2016, the Sing Consortium was created to comment and develop policy and process on genomic research and with Indigenous communities. And so from this consortium, we've been able to publish a number of papers that have been changing the narrative on how genomics research is done. So in 2018, for example, we published a paper on titled Advancing the Ethics of Paleogenomics, where we emphasize that ancestral remains should not be treated and regarded as artifacts, but as human relatives who deserve respect. We also developed a framework for genomics research with Indigenous communities that I will talk about later in the presentation. And so it seems like with the workshop and with the Sing Consortium, we're having an impact on science. And so just as a take home, with the Sing workshop, we're changing the practitioners and practice of science by increasing the number of Indigenous scientists that can serve as role models, and also changing the practice of science by indigenizing the practice of science and doing science in a way that fits with Indigenous culture and ideas. So this will be my only plug for this presentation. If you know of an Indigenous community member or Indigenous student who has an interest in science and genomics, they can go to www.singconsortium.org and sign up or fill out an application for our workshop that we're having this summer. This summer, depending on how it's going with recovery from the pandemic, it may be an online workshop or it may be in person. We're not sure yet, but pass the message along if you know of anyone. Okay, so now I just want to talk about community-based principles that we employ. I've been using community-based methods uh, for the past 15 years since I first started working with First Nation communities in British Columbia. When I started partnering with Dr. Jerome Sobolski, who's pictured here, when we began to work with First Nations on the interior of British Columbia around 2005. So Dr. Sobolski was a curator at the Museum of, at the time it was called the Museum of Civilizations near Ottawa. And he had been working in a community-based way with First Nations for about 30 years at that point. And so I learned a lot from him, and I learned partnership principles like respect, mutual benefit, and communication. And we would do things that that were not done in the case studies that I talked about earlier, where we would go back to the communities every year and report our most recent results and ask for feedback on those results to work towards a co-production of knowledge with the communities that we were partnering with. We also, so as I began to use community-based methods and time has gone on, my methods have evolved a bit, and the SYNC consortium and ended up publishing a framework on methods for genetic research with Indigenous communities. And so now I tend to use this framework as my basis. And here's a diagram of the framework where the center of it is the Indigenous community, and the sovereignty of that Indigenous community is at the center of the research. And so that's always important to keep in mind. So let's see. So for this research, there are 4 different principles that we like to employ: equity, respect, reciprocity, and beneficence. And then for the framework, we again like to keep transparency by keeping open communication with the partnering communities, have components of capacity and cultural competency, and then also we work closely with communities before we disseminate results to make sure that the language and what we're saying is agreeable to everyone in the partnership. So just in thinking about community discussions with the work we're doing now in British Columbia and Southeast Alaska, we worked to visit the communities in person often, and coming from Illinois, that these days means usually annually. And so here is a flyer from our community partners in Metlakatla First Nation. And so it gives some of the information on when we're going to be there, and then community members come, and I or a research team member will give a presentation and receive feedback from community members and participants on the results that we're seeing, and any ideas on where to go next with the research. These days, when we can't travel because of things like the COVID-19 pandemic, it's great that we have technology that we can use to visit the communities virtually using things like Zoom. And so here's a screenshot of me Zooming with our Metlakatla partners, Barbara Petzold and Joycelyn Mitchell, and then also another researcher that is these days a visiting scholar at Sealaska Heritage Institute, Dr. Alyssa Bader. And what's nice about this method is that the meeting is being recorded, and so other community members at Metlakatla First Nation can view the recordings and either get in touch with us directly or contact Joycelyn or Barb to pass along any questions or things we need to know about. The other thing that we like to do in order to make sure that we are transparent with the research is we like to invite community members to the University of Illinois and to the lab to see the research themselves and to see the labs and how we're doing all of the research. So not all community members can come visit the University of Illinois. So what we've been doing lately is we've been developing a video to show exactly what happens in the lab. So anyone in that community can see the process. And so with these videos, we make sure that we're not using human remains, we're using, in this case, salmon remains to do the DNA extraction and show the process. The hope is that at some point we will get to where technology advances enough to where we can actually take what's called these USB sequencers to the community, and we can actually do the DNA sequencing in the community with these portable DNA sequencers and a laptop. And so in that case, all of the research can be done at the community and not having samples taken from the community to a university that may be distant and far away. So with our partnerships on the north coast of British Columbia, we started working with co-Simpson communities around 2009. And so here is an aerial view of the city of Prince Rupert. But archaeological excavations in the Prince Rupert Harbor area have been going on since the mid-60s, up until recently when there was a port expansion and there was an archaeological excavation before the expansion was built in. And so overall, there are a number of ancestors that have been excavated from this region, over 250 ancestors from a number of different archaeological sites at ancient villages. And nearly all of these ancestors are currently at the university or the Museum of Civilizations. I think it's called the Museum of History now, near Ottawa. And these ancestors range from living about 1,000 years ago that are housed at Ottawa to 6,000 years ago. And so with the permission of the partnering communities, the co-Simpson, we were able to take samples, either a tooth sample or a small piece of bone, in order to do the paleogenomic analysis that we need to do, which is destructive analysis. About 0.2 grams usually is what we use. So with the permission of the communities, we were able to take the samples from the ancestors, and also we took saliva samples from present day community members in order to do some of the research that I'm going to be presenting here. Just to give you a sense of the geographic region, here's a map of the Prince Rupert Harbor region. Here's Prince Rupert. The Metlakatla First Nation is located here, and the Laqualams First Nation is located up here. I'll be talking about ancestors from Dodge Island and Lucy Island, that is off the coast from the Prince Rupert Harbor. But there are a number of ancient villages where ancestors were excavated over the years that we were able to analyze. And then if we just zoom out a little bit more, we can see that Prince Rupert is in this box right here that I just showed a slide on. But Southeast Alaska is very close by, right here. And so today they are in different countries, but pre-contact, this whole region was, all of these communities were close to one another. Okay, so two things that I wanted to mention before moving on to the paleogenomics projects. And the first thing is that there have been rapid advances in computational genomic analysis and paleogenomic analysis over this last decade. And so prior to this last decade, before 2010, let's say, we were really limited in the amount of DNA that we could analyze from an ancient individual. This right here is a schematic of the mitochondrial genome or mitochondrial DNA that traces the direct maternal line of an individual. I'll talk more about that in a bit. But before 2010, we were just able to analyze this 1,000 base pair or 1,000 units of DNA in an individual. And we weren't able to look at the nuclear genome at all, which is where most of your DNA is found, over 3 billion units as compared to 16,000 in the mitochondrial genome. So before 2010, the nuclear genome was pretty much off limits. But then after 2010, there have been such massive leaps and advances in technology that these days in my lab, if the DNA is preserved in an ancestor or an ancient individual well enough, we can analyze and sequence the entire nuclear genome, as well as we routinely sequence the mitochondrial genome along with it. And so there have been major advances over this past decade, which provides us more information to allow us to say more with this type of research. The second thing I wanted to point out is that this is a very large team effort. It's me and researchers that come through my lab. Here is a photo a few years ago of members of my lab. It's us along with our partnering Indigenous community members. So here is a photo of some of the members of the Metlakatla First Nation or folks that work for the Metlakatla First Nation. Here is researchers from the University of Illinois visiting Sealaska Heritage Institute with members from this institute. We have collaborators in other universities like Dr. Michael DeGiorgio, who is our computational genomics expert and is doing the cutting edge research on computation. Other researchers like Dr. Brian Kemp, who was the original researcher who led the DNA researcher or the DNA research on Shukaka, and others. So it's a very large effort that takes lots of time in order to do this type of analysis. And it's the effort of everyone involved that allows it to happen. And so I wanted to make sure to point that out. Okay, so for the first project that I'm going to discuss, it's where we were able to take a time transect of exomes of First Nation individuals near Prince Rupert Harbor. And so in partnership with the Metlakatla First Nation, we were able to take DNA samples from ancestors ranging from about 1,000 years ago to 6,000 years ago, and then also take DNA samples from living community members. And we were able to sequence the exome of these individuals. So some of you may be asking, what is the exome? So the exome is the part of the genome or the part of your DNA that codes directly for proteins. And so controls for our traits, whether they are physical traits or behavioral traits or traits that have to do with health. These are all part of the exome. And so in essence, it's we sequenced in the ancient individuals, the ancestors, about 60 million base pairs or 60 million units from 25 individuals, and then about 25 individuals from living community members from the Coast Tsimshian. In this research, after sequencing the DNA, one of the first things we want to do is to make sure that we are actually looking at authentic ancient DNA when we're talking about DNA from the ancestors, because there are possibilities of DNA coming off of a researcher or coming from lab equipment or from the reagents that we use that contaminate the ancient DNA with modern contaminant DNA. And so we want to make sure that we are using authentic DNA from the individuals that we expect to analyze. And so we uh, completed an analysis where we were able to create a tree, and in a tree, the closer two groups are to one another, the closer the genetic affinity. And once we took into account the impacts of European colonization, we were able to see a very close genetic affinity between the ancestors at Prince Rupert Harbor and Coast Tsimshian participants. And so this gave us confidence that we were actually looking at authentic ancient DNA from the ancestors. The other way to show that we're looking at authentic ancient DNA is because after an individual dies, the machinery in their body that keeps their DNA preserved breaks down. And so bacteria and other microbiota can start to break down the DNA and damage it. And so usually when you're working with ancient DNA from ancestors, it will be damaged. And we can look at a specific type of damage called deamination. And so when we look at deamination in our DNA sequence, we are able to see it and show that we actually do have DNA damage, which means that the DNA is coming from the ancestor and not some kind of modern contaminant. And so this was also relieving to see because we were able to show that the DNA results from the ancestors make sense. And it also shows that DNA damage, which demonstrates that it is authentic and not contamination, which has been a problem in ancient DNA research in the past. So once we were able to verify and do a quality control of our data, we can then start doing the analyses that we wanted to originally plan and are more excited about doing. And so the first thing we did is we wanted to see how, with this ancient population through time from 6,000 years ago to present day, what kind of demographic model fit that data the best. And so we ran a number of demographic models, and the one that fit the best is this one I'm going to show you right here. And the major component of this demographic model is that around 175 years ago, from the DNA data, the genomic data alone, we were able to show that there was a major reduction in genetic diversity. And that usually means a reduction in population size after European contact around 175 years ago. And this is from historical records, seems to be about this time when we had, there were smallpox epidemics on the north coast of British Columbia. And so we were able to see this similarity from the genomic data with historical records. And so that was interesting to see. The next thing we did is we completed a selection analysis. And what that means is that we were able to take a look at how the environment is influencing the DNA and the genome of the individuals that we were analyzing in the past as well as today. And so when we did this selection analysis, we were able to see these signals of selection on genes. And the strongest signal that we saw was on a gene called HLA-DQ alpha 1 that is associated with immune-related function or associated with infectious disease and fighting off infectious disease. And so this was really interesting to see this pattern of selection on this genomic region related to the HLA-DQ alpha 1 gene. The next thing we did is we ran models to see how this selection or how the environment changed pre-European contact from post-European contact. And the model that seemed to fit our data the best is this model right here that showed that there was a change after European contact for negative selection, or after Europeans arrived, they changed the environment dramatically by bringing pathogens and other changes that had a selection pressure on genes, in this case, an immune gene of individuals in indigenous communities. And so we can see that the impacts of European contact had potentially a strong effect on genes, in this case related to immunity for indigenous peoples on the Northwest or on the North Coast of British Columbia. So just to summarize this slide, from the genomic data from this study, we were able to identify a large population collapse following European contact. And then we were able to identify evidence of selection or on immune-related genomic regions, or how the environment changed the genomes of indigenous people after Europeans arrived on the North Coast of British Columbia. Okay, so now I'm going to move on to another study where we were able to identify, the goal was to identify DNA links or genomic links between present day community members and ancestors. And so this research was led by Dr. Yinkyu Kui, who spent some time in my lab and is now a professor at Jilin University, along with Dr. John Lindo, who did his dissertation in my lab and was able to do his research and is now a professor at Emory University. Okay, so in this research study, we focused on the mitochondrial genome. And with the mitochondrial genome, we are able to look at the mother's maternal lineage. And so here is a figure that describes the mitochondrial genome and how it is inherited. So here is a family tree, and here, let's say if you're a circle, you are female. If you're a square, you are male. And if you have a horizontal line, that means these individuals were married or they produced offspring through a vertical line. And so these are generations as you go back. And so the mitochondrial genome traces the maternal line, or your mother's 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 mother. And so if you go back one generation, it traces one ancestor, your mother. If you go back 2 generations, it traces the maternal line of your mother's mother, but not these other 3 ancestors. If you go back 4 generations, it traces back your mother's mother's mother, but not these other ancestors, right? And so it gives you this view of this single line and the genetic relationship of individuals along that line. And so when we looked at ancestors in the region and compared them to DNA from, or mitochondrial DNA from present day community members, we found a few interesting links. The first link was from an ancestor that was who lived on the Lucy Islands off the coast of British Columbia. And this ancestor lived about 5,000 years ago. And then this ancestor had a mitochondrial genome that was the exact same mitochondrial genome of an ancestor that lived on Dodge Island in the Prince Rupert Inner Harbor, who lived about 2,500 years ago. And this ancestor had the exact same mitochondrial genome of a living Coast Tsimshian community member, which was surprising and somewhat unexpected. And so that was a really interesting find. The other DNA link that we found was more of what we expected. This was a DNA link where we had an ancestor who lived about 5,000 years ago on Dodge Island in the Prince Rupert Inner Harbor, and had a similar, very close related, but not exact mitochondrial genome to living community members in the Coast Tsimshian, but also neighboring communities like the Haida and the Niska. Right? And so with these results, here is a popular news story where we have a quote from Barbara Petzold, who works for the Metlakatla Treaty Office. And she says, having a DNA link showing direct maternal ancestry dating back at least 5,000 years is huge as far as helping the Metlakatla prove that this territory was theirs over the millennia. So this is a nice quote that shows how this research might be helping the partnering community that we are working with. But I want to make clear that in this study, 2 points. One is that we weren't just looking at DNA results alone. We were looking at DNA results and DNA information along with archaeological information that also went back about 5,000 years. And then that along with indigenous knowledge and oral histories. And so it was the 3 sources of information coming together that we were able to present this scenario. And so we just wanted to make that point. The other point I want to make is that the comparative database for mitochondrial genomes on the north coast of British Columbia or northwest coast of North America in general is very small. There are lots of First Nation communities and Indigenous communities that have not contributed to the database. And so some of these mitochondrial genomes may be found in these other present day communities, but we just don't know because they are not in the database. So that's important to keep in mind. Okay, there's a third DNA link that I wanted to talk about, and this one is also very interesting. So there was another ancient individual, an ancestor who lived on the Lucy Islands at about 6,000 years ago. They did not have a mitochondrial genome that we found in any of the living descendants that were sampled and sequenced for the mitochondrial genome. But there was a link to another ancestor that lived north at about 10,000 years ago on Prince of Wales Island. Shukaka is what this ancestor ended up being named. So we know this because in 2007, Dr. Brian Kemp and myself were part of the DNA team in the analysis of Shukaka, where we were able to identify the mitochondrial genome by sequencing this small portion of the mitochondrial genome. And we were also able to identify the Y chromosome of this ancestor. And so we were able to find this connection between Shukaka 10,000 years ago and the individual labeled 939 or called 939 on the Lucy Islands at about 6,000 years ago. And so we were also able to look at the Y chromosome and see this relationship by looking at, again, instead of the mitochondrial genome, which traces the direct maternal line, the Y chromosome, which traces the direct paternal line or your father's 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 father. Right? And so from the 2007 study, we were able to present information on the mitochondrial DNA and the Y chromosome of Shukaka. And this was, again, remember, this was before we had this leap in abilities for sequencing DNA, right? And so I just briefly wanted to, I'm running late on time. I briefly wanted to mention the partnership on Shukaka because it is remarkable. So this ancestor was identified in 1996, the same year as the ancient one. And unlike the ancient one, as soon as it was identified, the researchers quickly communicated with the local communities in order to work with them to see if scientific research was a possibility, and then collaborated with the local indigenous communities on the research all the way through to when this ancestor was reburied in 2008. So it was very different from the ancient one in that it was a collaborative relationship in this case, as well as opposed to an antagonistic relationship as with the ancient one. So what we ended up doing after seeing this mitochondrial DNA connection between 939 on Lucy Islands and Shukaka is that we again got permissions from the local communities as well as SHI to see if we could complete a study of the nuclear genome on individuals on the Northwest coast. So this would be Shukaka at 10,000 years, 939 at 6,000 years, an individual called 302 at 2,500 years ago, and an individual called 443 at 1,750 years ago in the Prince Rupert Harbor area. And so we were able to look at this time transect from a nuclear genome point of view. And so we ended up being able to get really good nuclear genome information and DNA from these individuals that were a little bit younger. The Shukaka ancestor is older at 10,000 years, and so we were able to get enough DNA information that bordered on the cusp of being informative. And so what we ended up finding, briefly, for the nuclear DNA inheritance, we're looking at information from all of our ancestors into an individual, unlike the mitochondrial DNA in the Y chromosome. So going back to what we found with Shukaka is that we were able to identify that with all of the analyses that we completed, that Shukaka was from the same general population and along the same nuclear lineage as other individuals on the Northwest coast. So 939, 302, 443, Shukaka, and a present day individual all show that they were on the same genetic lineage in North America. And this was opposed to other ancient individuals like the Anzac individual, who lived about 12,000 years ago in Montana, has a lineage that goes to descendants who are now living in South America. And the ancient one was also part of this lineage. And so in the end, we were able to identify that there was local genetic continuity to present day with Northwest Coast individuals from Shukaka all the way to present day. So just to summarize this part of the research is that we were able to identify regional genetic continuity on the Northwest Coast of North America and the presence of genetic population structure back at 10,000 years ago during the late Pleistocene, just after the last glacial maximum. Okay, and with that, I want to say thank you, and I am happy to take any questions. Here are my acknowledgements.
Well, thank you very much, Dr. Malhi. There are a couple questions that come up. One is that this person comments, I love the language you use when describing your data samples as ancestors. Do you get pushback from the academic community because it suggests a relationship between the researcher and the data sample that contradicts the objectivity standard?
Right. Yeah, so with this notion of objectivity, I push against that idea because it seems, as I showed earlier, Western science is dominated by one group of people, right? Basically, uh, European descendants, and so what seems to be objective is actually just a norm for that community, right? And it's as subjective as everything else. And so I push back against that idea of objectivity because there is subjectivity in science and the way we not only interpret our results, but the way we design our studies um, uh, and what questions we ask. Um, and so I, I, I guess I may get pushback, and if I do, it's not strong pushback.
Another question is, I wondered if you could go back to your table showing the lineal relationship between Shukaka and modern day Northwest Coast tribes,
Sure.
and explain the relationship between that evidence, that information, and the relationship to lower 48 tribes, and all the way down to Tierra del Fuego, which, as I understand it, there were some connections there, and even to the Mound people in the Midwest, eastern side of the U.S.
Uh,
Um,
so this slide, or was it this one that you wanted me to?
that one.
Okay.
Can you connect that to the lower, like South America? Could you discuss those relationships? Because I understand that's what uh, was pretty important for the showing of a connection that related to ancestry for repatriation purposes, that it
Right.
was Native American as opposed to Asian, which is what some of the scientists were saying about Kennewick Man.
Mhm. Yeah, and so I mean, at the time that we did the study, there were very few late Pleistocene ancestors that were analyzed through paleogenomics. And the ones that were all showed this uh, connection on a genetic lineage that wasn't just in North America. It was in North America, but then also you would find the same genetic lineage in South America. And so this um, lineage was known to go back into the late Pleistocene, whereas the lineage that Shukaka ended up being on was related to these other Northwest Coast individuals. This one from the Lucy Islands at about 6,000 years ago. These from the ancestors from the Prince Rupert Inner Harbor um, at about 2,000 and 1,000 years ago, and then uh, community, present day community member on the Northwest Coast. And so it wasn't known that this lineage actually went back into the late Pleistocene. And so when we first analyzed Shukaka, the data was, we were trying to do, we had two options that seemed to not be rejected, the models that were not rejected. One was that Shukaka was on a lineage that split off before these two lineages and was part of its own lineage. The other was that Shukaka was on this lineage that traced down to other descendants on the Northwest Coast.
Hmm.
And so through simulations and other analyses, we ended up being able to demonstrate that it was more than likely that Shukaka is on this lineage that traced down to the Northwest Coast.
Well, thank you. That's very interesting. Um, Another question is, how do the Northwest Coast genetic relationships relate to major geological events, like the little ice age, in terms of geographic distribution? Can migrations and such be correlated with those events?
So there are ways to actually look at genetic dating and using genetic diversity to identify a time period of when an event actually took place and see if that correlates with some kind of geological event. But the closer you get in time, the less reliable those genetic dates are. And you also need a large sample size in order to constrain the variation or your error estimates on those genetic dates. So I guess I would say that it's possible to do that type of work, but during the Holocene, when there's a lot of interest in doing this type of correlation, in North America, not much work has been done because you need um, such a large sample. And in order to do it using the nuclear genome, we just haven't been able to do it with paleogenomics because the field is so young. And we're just now starting to change our relationship by partnering with indigenous communities instead of doing what scientists have done before and just doing the research without consulting with communities.
Alrighty, well, thank you again. This has been very enlightening, and it's really good to see it expressed this way with your charts and all. SHI invites viewers to return for our next lecture by Steve Brown, entitled Evolution of Northwest Coast Art, which will be broadcast on Tuesday, January 19th at noon. We have a link below the YouTube video for a survey we hope you will take. This will help us to continue improving our lecture series and also allow our funders to measure the impact of the program. And it doesn't take very long. SHI has started construction of its arts campus in downtown Juneau, which will expand opportunities for education and art. If you are interested in making a donation to the arts campus, please visit sealaskaheritage.org/campus. Thank you all again, and we'll see you on Tuesday.
Thank you. It was fun.